Hello everyone, myself Nadini from Maharani Science College for Women, Bengaluru. In this session, I shall be covering the introduction of nitrogen metabolism. Nitrogen is an important inert gas which is present in all the living beings. They make universally occurring element nitrogen, and we all know in any cell more than 10 to 12% of the proteins are present, which are made out of amino acid monomers. So amino group involves the nitrogen. Nitrogen makes a very important component of even the chlorophyll, and also very many important substances which are present in the body, like hormones, vitamins, etc. So these proteins are the building blocks of the protoplasm and are made up of nitrogenous substances. The amino acids in turn are synthesized when inorganic nitrogen of the environment is converted into organic nitrogen. This is the important aspect of nitrogen metabolism. Nitrogen metabolism is a very important process next to photosynthesis and respiration in plants. Here, atmospheric nitrogen is inert inorganic nitrogen gas. In that form, the plants cannot utilize nitrogen, so we need to see for the simpler forms. The plants they themselves are going to convert this atmospheric inert nitrogen to the nitrates, nitrites, and ammonium ions. And how exactly those things are done even in nature, in the chemical, physical, and also natural means. So that is the importance of this topic, nitrogen metabolism. Even organic nitrogen is also seen in the dead and decaying organic matter. And thus nitrogen makes the major part of our atmosphere. Approximately 78 to 80% the nitrogen will be present. It is important component of chlorophyll, amino acids, and proteins, stimulate root growth and also root development, and it is going to help in the uptake of cations, encourage vegetative growth, impart dark green color to the plants, improves the quality and succulence of leafy vegetables and also fodder crops. Nitrogen is an asphyxiant gas, is a non-toxic and minimally toxic, which reduces or replaces the normal oxygen. That is the meaning of asphyxiant. So it is going to reduce or displaces the normal oxygen concentration in breathing air. Breathing of oxygen depleted air can lead to death by asphyxiation or suffocation, because most of asphyxiant gases are inert and they are odorless. The processes in the nitrogen cycle is to transform the nitrogen from one form to another form. As atmospheric nitrogen is highly inert and it is made out of triple bond, so it has to undergo the reduction process to get the simpler forms like nitrate, nitrite, and ammonia. And later it is assimilated by the plants. Many microbes which carry out this process of nitrogen fixation. So for example, nitrogenous wastes in animals, in urine and also fecal matters will be broken down by nitrifying bacteria in the soil, and thus it is going to produce the fertile humus, and that will be assimilated by the plants, taken by the plants by the nitrifying bacteria. So there are three important steps, that is nitrification, nitrogen fixation, and also the assimilation of the nitrogen, and also denitrification, that is the conversion of the organic nitrogen into once again giving back the molecular nitrogen to atmosphere, that is denitrification process. All the three processes are involved in the nitrogen cycle, which is an important biogeochemical cycle. So along with that, we are going to discuss in nitrogen metabolism topic the biosynthesis of amino acids, which are the building blocks of proteins, and which makes the major source of nitrogen in the organic as organic form. So let us see the first topic under nitrogen metabolism, that is sources of nitrogen. Nitrogen makes the major source in the atmosphere and also in the soil in the following forms. In soil, we can see that the nitrites, and also nitrogen, which is present in the oxidized state, and it is inorganic. And also one more inorganic form of the nitrogen is ammonia nitrogen, which is present in the reduced form. And two types of organic nitrogen forms are amino acid in the body of the insects, so in the body of the insects, also we can see the one more organic form where we can see the insectivorous plants which are going to depend on the insects. They will grab and trap the insects for their nitrogen source, for their nitrogen source. And also the molecular nitrogen which is present in the atmosphere. These are the important sources of nitrogen. As been already pointed out, except for few prokaryotic organisms like bacteria and cyanobacteria, the rest of the plants contain they need to obtain the nitrogen from the soil source. They must depend on the soil, they must depend on these organisms to get the nitrogen fixed into usable or assimilatory forms. So now let us see one by one all these sources, a little description about these sources. First one is the inorganic source, that is nitrate nitrogen, NO3 minus, and also NO2 minus, that is nitrite ion. The decomposition of the organic matter obtained due to the death of plants and animals result in the formation of nitrate ions in the soil. Nitrate ions are negatively charged and are loosely held to the soil missiles. Before being taken up by the plants, the nitrate ions have to be reduced to ammonia. Plants cannot take nitrate ion as it is, so they need to get reduced in the presence of nitrogen reductase enzymes or nitrogenase enzyme, and thus the chemical reaction, the overall chemical reaction can be seen here. Even the reduction takes place within the cells of the plants and needs the participation of adenosine triphosphate besides the hydrogen donor. The overall reaction is nitrite ion in presence of reduced ferredoxin, 2 electrons and also 2 protons utilization. You can see the formation of the NOH, and then the same thing repeats to give the hydroxylamine, and also in the next step we can see the formation of the ammonium ion. And also nitric acid undergoes reduction to produce the ammonia and water. In order to reduce the nitrate to ammonia, we need 8 hydrogen, that is hydrogen atoms are required, which are provided as a source from the respiration and photosynthesis. According to Evans, Nessen and Nicholas in 1957, the following are the stages of the nitrate conversion. We can see the nitric acid forming nitrate ion and also the hyponitrite and hydroxylamine and finally into ammonia. Various enzymes are involved in this process, that is nitrate reductase, nitrite reductase, nitric oxide reductase and hyponitrite reductase. These are the enzymes which are involved in the completion of the conversion of the nitrate to ammonia, that is the usable form for the plants. And next inorganic source of nitrogen is ammonia nitrogen. Ammonium ions are the cations, so they are very tightly held to the soil micelles. This is first converted into nitric acid by soil bacteria. The process is called as nitrification. So nitric acid is first formed, that is the nitrification, the conversion of the major complex forms of nitrogen to nitric and nitrate ions is the nitrification. Ammonia is made available to the plants through nitrate reduction or metabolic degradation of proteins. Even urea is also a good source of ammonia. Urea is an important fertilizer that is found in the soil that combines with water to form ammonia, and carbon dioxide is liberated. So third source, that is the organic nitrogen source. Plants and animal debris after the death of plants and animals, and also the excreta and other decaying organic material makes the major organic source of nitrogen. Organic nitrogen is not directly absorbed by the roots of the plants except for urea. Only for a few fertilizers like urea and uric acid, all other sources are not actually directly grabbed by the plants. So these organic molecules containing nitrogen which remain adhered to the soil particles, they break down into amino acids first, followed by the oxidation of ammonia and organic acids. Ammonia thus released immediately gets oxidized to nitrate, which on absorption by plants again gets converted into ammonia. Once again it will get converted into ammonia after the absorption by plants. When urea is the source of organic nitrogen, it is first hydrolyzed by the enzyme urease, resulting in the production of ammonia and also carbon dioxide as the byproduct. We can see the last source of nitrogen, that is the major source, that is molecular nitrogen. This is an inert but occupying the bulk of the atmosphere. It is not available to the plants in the gaseous form, as the plants do not have the genetic potentiality to use this form of nitrogen. So gaseous nitrogen has to get converted into nitrate and nitrite before it can be used by the plants. So how exactly this the conversion of the molecular nitrogen to nitrate and nitrite takes place? That is by the nitrogen fixation. So nitrogen fixation is the process by which the atmospheric nitrogen is converted by either natural or an industrial means to form the nitrogen such as ammonia, which is an usable form or consumable form for the plants. In nature, most nitrogen is harvested from the atmosphere by microorganisms to form ammonia, nitrites, nitrates, ammonium ion, etc. that can be easily used by the plants. Nitrogen fixation, again we will discuss under three important headings. That is industrial or chemical fixation, which causes free nitrogen, which is relatively an inert gas which is present in the atmosphere, which will combine chemically with the hydrogen molecule under high temperature and atmospheric pressure, higher pressure up to 300 bars, that will convert into finally into ammonia, nitrites and nitrates. That is the chemical fixation. Under ordinary conditions, nitrogen does not react with other elements. So that's why we need to provide higher temperature and pressure. And one more important thing during last 19th century, during the world wars, there was an important need for the nitrogen source for to feed the agriculture sector. And at that time there were fertile soils which are made out of the coal and also the coke. While the farming or while producing the coal and coke, the liberated obtained nitrogen was importantly harvested as a fertilizer. And also naturally occurring chemicals such as sodium nitrite, that is popularly called as chili saltpeter, and ammonia. These substances were very short for to feed to the nitrogen which is required as a fertilizer to the growing plants. So nitrogen is also found in the nucleus of every living cell as one of the chemical components of DNA. As I already told, it is an important component of nucleic acids. So nitrogen makes the very chief ingredient for the plants in the agriculture sector. So all the available, naturally available nitrogen sources like chili saltpeter, they are not at all sufficient. So we need to think for the production of the usable forms of nitrogen through artificial nitrogen fixation. So various types of nitrogen fixation will be our next topic of importance. See here, nitrogen fixation is broadly classified into two major types, that is artificial and natural. In natural nitrogen fixation, we study two important types, that is physical and biological. Physical will be the lightning and also UV rays related nitrogen fixation. And very importantly, very major one that we study here in nitrogen metabolism is the biological nitrogen fixation with the help of non-symbiotic and also symbiotic microorganisms. So nitrogen fixation is totally how much amount of nitrogen that is fixed in the atmosphere through various processes is 10 to the power of 12 grams per year, or to put it in other way, 10 to the power of 6 metric tons per year. So there are different types of nitrogen fixation. Amongst that, non-biological nitrogen fixation, in that industrial nitrogen fixation will contribute about 50%, and also the nitrogen fixation through combustion of the natural gas and also fuels from the automobiles, that contribute 20%, and lightning 10%. So totally non-biological nitrogen fixation is up to 80%. And coming to the biological nitrogen fixation, biological that is through the agriculture land, about 90%, forest and non-agricultural land 50% of the nitrogen gets fixed, and through the sea, that is how exactly the processes involved with the microorganisms, that is 30%. Overall biological makes 175%. So the total requirement is almost met by the biological nitrogen fixation, and only 1/4 and less than 1/4 will be taken care by the non-biological nitrogen fixation. Let us see what are these non-biological nitrogen fixation types. See, in non-biological nitrogen fixation, the first one makes the industrial or chemical nitrogen fixation. Here, under specially created industrial conditions at very high temperatures up to 450 to 500 degrees centigrade, and atmospheric at pressure of about 300 atmospheres, higher pressure, we can see the combination of the nitrogen, inert nitrogen along with hydrogen. Okay, that is going to give the, I will just enlarge this picture. So we can see in the huge furnaces, this is the instrument that is used in the chemical nitrogen fixation, and it was called, this process is called as a Haber-Bosch process of nitrogen fixation. Here nitrogen and hydrogen combines to give ammonia, to produce ammonia. This process is called as Haber-Bosch process. Here a huge amount of energy requirement is there. See the delta G, that is the enthalpy, the amount of energy requirement is minus 22 kilocalories. So that much amount of energy is utilized in this process. So it is a laborious process, the chemical non-biological nitrogen fixation. Coming to the physical non-biological nitrogen fixation, that makes the combustion, that is the important step. Nitrogen fixation here is associated with the activities of humans. The internal combustion of the engines of automobiles burn fuels under conditions of higher temperature and pressure, which favors the oxidation of the nitrogen gas to nitric oxide. An important air pollutant and source of fixed nitrogen, even though the nitric oxide NO is an important air pollutant, but the atmospheric nitrogen gets fixed. The combustion of coal and other organic fuels also result in the formation of nitric oxide through both the fixation of nitrogen gas and oxidation of the organic nitrogen of the fuel. So thus the combustion makes the chief source of non-biological nitrogen fixation, but it contributes very less to the total amount of nitrogen requirement. Next one will be the natural process, that is through lightning. It is a natural way nitrogen in the atmosphere can be transformed into plant usable form. The process is called the lightning nitrogen fixation, or it is the natural nitrogen fixation by lightning. Each bolt of lightning, lightning that happens carries electrical energy that is powerful enough to break the strong bond between the two nitrogen atoms, that is N triple bond N. So that bond, breaking of that bond is easily done with this huge, enormous amount of electrical energy in each bolt of lightning. Here, whenever that bond has to break down, then we can see nitrogen combines with oxygen to form the nitric oxide. This nitric oxide again combines with one more molecule of oxygen and undergoes oxidation to give nitrogen peroxide. Later, nitrogen peroxide mixes with rainwater and it is shed down as the nitric and nitrous acids, can be called as acid rain. So natural nitrogen fixation. Nitrogen is fixed or combined in nature as nitric oxide by lightning and also UV rays. So whenever there is lightning, in presence of UV radiations, more significant amounts of nitrogens are fixed as ammonia, nitrites and nitrates. Much more requirement is met only through the biological means, that is with the help of microorganisms. More than 90%, more than 90% of the overall nitrogen fixation is affected only through the biological means. So we must be aware of those microorganisms which are contributing to this maximum amount of biological nitrogen fixation, that is meeting the demand of the plants which can accept this nitrogen source, fixed nitrogen source, and they are growing in all agricultural crops and horticultural crops. So we can see the contribution of these microorganisms. First one, free living non-symbiotic bacteria. It is free living and also non-symbiotic bacterial population. Those include cyanobacteria, that is blue-green alga, Anabina, Nostoc, and genera such as Azotobacter, Baezieringia, and also Clostridium. These are the non-symbiotic free-living bacteria and blue-green alga. And next one is the mutualistic, that is symbiotic bacteria such as Rhizobium associated with leguminous plants, and Azospirillum species associated with cereal grasses. And other examples of these microorganisms are also there. Let us see a bit later. I would like to tell only few points about industrial nitrogen fixation history glance. See, during the 19th century, very much need for the nitrogen sources were there. So nitrogen materials have long been used in agriculture as fertilizers. That we all know, NPK makes the major fertilizer. In that, nitrogen deficiency will cause the plants look very pale in color, decolorization, etc. So it is increasing need for the nitrogen, fixed nitrogen for the plant sources. And accordingly, ammonia released in making the coke from coal was recovered. This particular ammonia which is released while making the coke from coal, that ammonia is utilized as fertilizer, and also the deposits, natural deposits of sodium nitrate, that is nothing but chili pepper, that sodium nitrate is also acquired, and all these supplements were fed to the growing need of the nitrogen, fixed nitrogen. At the same time, during those times, this chili pepper was very much used to make the gunpowder, and thus the natural deposits of this nitrogen compounds were very much diverted in the production of the gunpowder, and thus these demand cannot be met, where the fixed nitrogen source demand cannot be met through these two sources, naturally occurring sources, and also through the combustion of the coke to produce the coal. During that process, the ammonia released was very much used as a fertilizer, but they could not meet the demand. So three most other productive approaches were done, where direct combination of the nitrogen with oxygen and also nitrogen with calcium carbide and combination of nitrogen with hydrogen were all done at higher temperatures and pressure. They try to get the assimilatory forms of nitrogen, gaseous nitric oxides, etc. And also this nitric oxide is then chemically converted into nitrates, and this experiment was done in electric generators were in use near Niagara Falls of New York, and also there also they used the technique, that is electric arc technique. It was harvesting lots and lots amount of energy. So they are very laborious processes, and at the same time they could not meet the demand. Even some electric arc or arc method commercial production of nitrogen were done, but all those were very costly and inherent inefficient processes. They could not meet the demand. One such method Okay, one such method approach was to use the first approach. One such method to get the nitrogen, that is the reaction of nitrogen with calcium carbide at high temperatures to form calcium cyanamide, which hydrolyzes to ammonia and urea. The cyanamide process was utilized on a large scale by several countries before and during World War One. But later in 1918, the Haber-Bosch process had rendered it absolute. And thus, finally, the Haber-Bosch process was standardized to get the complete production of the chemical nitrogen fixation, non-biological chemical nitrogen fixation. Thus, the Haber-Bosch process makes a very important amongst all those efforts that were done to meet the demand for the assimilatory nitrogen sources. Next, coming to the biological nitrogen fixation, once again the two major types, that is asymbiotic biological nitrogen fixation and symbiotic biological nitrogen fixation. Asymbiotic nitrogen fixers are the microorganisms. We can see a few of the microorganisms which are autotrophic, free-living aerobic bacteria which fix the atmospheric nitrogen using their heterosis. Very important here, heterosis. Heterosis are the specialized cells in the filamentous forms like nostoc anabena. These cells we can see very less amount of chloroplast. So since the chloroplast is very less, the amount of photosynthesis is highly reduced. Only photosynthesis system 1 operates where there is no lysis of water and thus no production of much oxygen. So heterosis is going to make a natural oxygen scavenging mechanism or the cell, which is very much required for the activity of the enzyme nitrogenase, which is involved in the nitrogen fixation. That is how almost all the filamentous blue-green algal forms which have the heterocyst, they are the very good source of nitrogen fixing microorganisms. Nostoc anabena, Tolipothrix, Alocera, and also Calothrix are the other examples for autotrophic free-living aerobic bacteria. And also non-heterocysts, blue-green alga also fixes atmospheric nitrogen, that is Gliotheca fixes atmospheric nitrogen, but the number is quite less in non-heterocyst forms. Then autotrophic free-living anaerobic bacteria. Autotrophic free-living anaerobic bacterial species like genera like Chromacium and also Rhodospirillum. They make the two important genera which fixes the nitrogen. And heterotrophic free-living organisms like aerobic under heterotrophs, that is Azotobacter. Very important. Azotobacter, in culture, it produces the extracellular polysaccharide which prevents the percolation of oxygen. So oxygen prevention itself is a major phenomena because it favors the activity of the enzyme nitrogenase, which works better whenever there is anaerobic condition. So that condition is created in Azotobacter. It is also a aerobic bacteria, but it prefers as the anaerobic phenomena to fix the atmospheric nitrogen. Azospirillum, Bergerichia are the other examples. of aerobic bacteria. And then coming to the heterotrophic anaerobic bacteria, Clostridium, Methanococcus, Disulfovibrio, purple sulfur bacteria, purple non-sulfur bacteria, green sulfur bacteria. These makes the heterotrophic anaerobic bacterial genera, which fixes the atmospheric nitrogen. Then last one, facultative bacteria, includes Bacillus and Klebsiella. Klebsiella pneumonia, which is a pneumonia causing organism, but it is a very efficient nitrogen fixing bacteria. Then coming to the symbiotic nitrogen fixing bacteria. What is symbiosis? It is association with other organism. For example, some plants like leguminous plants and also other genera of plants which makes the association with the bacteria and also the blue-green algal population, which is going to help in the nitrogen fixation. So symbiotic aerobic bacteria in root nodules of leguminous plants, very famous. Example is Rhizobium, Azorhizobium, Bradyrhizobium, Photorhizobium and Xenorhizobiums are the other examples. Rhizobium symbiotic association with root nodules of another plant, that is Parasponia, which is popularly called as Trema of Almaceae. And also actinorhizal plants, that is in the genera 23 genera of non-leguminous angiospermic plants, which are all dicot woody shrubs and trees, for example Casuarina, Myrica, Ulmus, etc. Associated with filamentous soil bacteria, that is Frankia, which is going to produce the orange colored nodules. Okay, Frankia belongs to actinomycetes. Without specific host, it fixes the atmospheric nitrogen. Very, very important feature. In case of Rhizobium, it is very much specific to leguminous plants, but this particular Frankia is a bacteria which is going to fix in 23 genera of angiospermic plants and also the species. Okay, also in the gymnospermic root of Podocarpus, it fixes the atmospheric nitrogen. So Frankia, which is going to produce very important in aldar plant, that is technically called Ulmus. Okay, it fixes the atmospheric nitrogen by producing the orange colored nodules. Next, coming to the leaf nodules in the Dioscorea, Psychotria, and also in Gymnosperm pavetta and Chromalia. And Anabina and Azolla, it's a very important association. Azolla is a phylum, it's a pteridophytic plant which is going to have the association of blue-green alga Anabina, and here and there in paddy fields we will find this Azolla, and we wrongly interpret Azolla is fixing the atmospheric nitrogen. No, it is inside Azolla the association of Anabina. So Anabina is actually fixing the atmospheric nitrogen without producing the nodules, hence it is a non-nodulating form. And also in Cycas, production of the coralloid roots in forming Coronoriza, so non-nodulating. So we studied so many important nitrogen fixing organisms. Let us see a few more pictures, slides in our next session about these organisms, and also how exactly they will fix the atmospheric nitrogen into usable forms. That we will study in our next session. Very importantly, the biological symbiotic nitrogen fixation. Let's summarize the topic. Today we studied the sources of nitrogen. The important source is the molecular nitrogen, which is present approximately 80% in the atmospheric gases. but because of its inert nature and asphyxiant nature, it is not easily accepted by the plants and also other organisms. When this particular nitrogen is obtained with the depleted oxygen level in the atmospheric gas, then the death can happen because of the reduced depleted oxygen due to suffocation, or also called as asphyxiation. And also we saw the important types of processes that are take place in the nitrogen cycle, that makes a very important aspect of this nitrogen metabolism. Those are nitrification, ammonification, and also nitrogen fixation and denitrification, nitrogen assimilation finally. So final denitrifying bacteria, which is going to send back the molecular nitrogen once again into the atmosphere, that makes the nitrogen cycle. And the important aspect of this chapter is nitrogen fixation, that we study in various headings, that is industrial or chemical, natural, physical nitrogen fixation, and also the biological. Chief one would be the biological nitrogen fixation using a symbiotic and also symbiotic bacterial populations. This completes this session, introduction to nitrogen metabolism. These are some of the references that we see. Happy learning. See you in the next session with biological nitrogen fixation. Bye for now. Take care. Good luck.